Okay, slide 21. This is the what's called cross-bridge cycling, or the sliding filament theory. It's hyphenated, and this is the best theory as to how muscle actually happens, and it all has to do with the myosin heads, or what are called the cross bridges. Myosin head has uh, binds to actin and has an ATPase on it, but once the myosin is moved out of the way, the myosin head immediately attaches to the actin. Has nothing to do with ATP. And then when the myosin head attaches to the ATP, it flexes. This is called the power stroke. And this causes the thin filament to slide past each other. Then, once the cross bridge has flexes and it has a different angle, versus 90 degrees goes to 45 degrees, and then the ATP binds to the myosin head. This detaches the head from the actin, and then ATP is burned. That means hydrolyzed, is burned, and the myosin head is recocked back to 90 degrees and binds to the next binding site on the next actin down the thin filament, and immediately binds and goes to power stroke. And this is called cross bridge cycling. Let me show you this in the next slide too. Okay, slide 22, same thing in picture format. Uh, Calcium binds to troponin, causes a conformational change in troponin. It changes shape and pulls the entire troponin tropomyosin complex, moves, and then it uncovers the binding sites for myosin on actin. Then, as soon as that happens, the cross bridge formation, the myosin head binds to actin. Then it immediately goes to the power stroke. The myosin head swivels toward the center of the sarcomere, pulling along the attached thin filament. This is called the power stroke. ADP and PI are released during this process. Then 3D, release of myosin head. ATP then binds to another ATP molecule, binds to the myosin head. As soon as that happens, it's hydrolyzed. In 3E here, the myosin head is reset. It's moved back up to 90 degrees, and now it's directly across from another myosin binding site and actin farther down the thin filament, and immediately binds and goes through the power stroke and the whole thing. And this goes on and on as long as this calcium binding to the troponin. And remember, every thick filament is surrounded by 6 thin filaments, and there's gazillions of these cross bridges forming, going to power stroke, and we're saying to pull the two filaments past each other, and cause the sarcomere to contract. Hence, it's called the sliding filament theory. So this is seems to be an odd way of muscle to work, but this is the best theory we have. It's really like a ratcheting mechanism. I think of it. I always use the analogy of a rowboat. That the water is the thin filament, the boat is the thick filament, and the oars are the cross bridges. And as soon as you're over the water, you put the oars in the water, the cross bridge attach, it goes to a power stroke. You take the oars out of the water, you burn ATP, you put it back further down, you put it again in the water. So it's just a ratcheting mechanism. And what that causes is this sarcomere to be shortened. So here's a relaxed sarcomere right here. We can see an H zone, and then here's a fully contracted sarcomere. The H zone disappears, and we have a complete overlap here of the two filaments. Okay, slide 24, relaxation. We've turned on a muscle cell. We've got it fully contracted. Now we got to relax it. So once the nerve signals stop arriving at the neuromuscular junction, acetylcholine esterase, remember, breaks down the acetylcholine. That's how we turn off an acetylcholine synapse. Active transport, this causes calcium pumps in the membrane of the terminal cisternae of the sarcoplasmic reticulum to resequester calcium by it binding the calsequestrin when it comes into the sarcoplasmic reticulum. Once the calcium is gone and dissociated from troponin, tropomyosin moves back over the binding sites for myosin and blocks the cross bridge formation. And the elastic filaments that tighten or connecting actually releases like a rubber band that's been stretched, releases and pulls the sarcomere open, and you end up with a relaxed sarcomere and a relaxed muscle. And even though every sarcomere shortens by a tiny microscopic amount, there are so many sarcomeres in a very long muscle cell that all of these little tiny shortening adds up to a macroscopic shortening in a muscle and tension generation by the muscle. Next slide, video we're going to go into muscle metabolism.